let's do it that way. That is good news. And another piece of good news, we managed to connect. So let's get the show on the road because we're perfectly on time. So thank you so much for joining us today, Ileana. We're super excited about the new microstructure and functional MRI techniques applied in neuropsychiatry, and a little bit about your background for everyone in the room and also live on YouTube. You have a dual degree in, brace yourselves, guys, physics and medical physics from France and Canada. What a double bill. And a PhD in physics from the University in Paris in France to work on the functional and diffusion MRI and microscopy at the single neuron level. Super exciting. And today we're going to hear about your latest research. I can't wait, so I'm going to give the floor to you right away. And as we just discussed, if you want to have questions answered straight away, raise your voice. And in case you can't see us, also literally raise your voice. Without further ado, Ileana, take it over.
Thank you so much, Stephanie, for the introduction and also for the invitation. I'm actually very, very excited to be presenting as part of these seminars today. And yeah, please feel free to speak up if anything comes to mind as I'm going through this through these slides. Okay, so just a little bit about the background of our field, which is microstructural MRI. So I guess the premise is that we need some kind of technique that allows us to do in vivo non-invasive microscopy. And so we have a lot of microscopy techniques out there that are absolutely outstanding for their own purposes. But unfortunately, none of these can really be applied to the living, breathing human, when actually from a clinical perspective, it would be very important to get information about pathology directly in the patient in a sort of non-invasive way, because this would really improve early detection maybe of disease and also improve the ability to do monitoring and treatment response in some kind of seamless way. And so the question that we asked ourselves, and really the rationale behind microstructural MRI, is can diffusion MRI provide such information to some kind of microscopy level, but in vivo and non-invasively? And of course, when we think of MRI, we realize immediately there is a huge gap in spatial resolution that needs to be filled, because our images basically have a spatial resolution of maybe 1 to 2 millimeters. When if we're thinking about microscopy and typical sizes of cells, I mean, human neurons, they are rather on the order of a few microns. So there is a huge gap there. And the beauty or the serendipity behind diffusion MRI is that it's somehow able to bridge that gap, because in a diffusion MRI experiment, we sensitize the signal to the mean square displacement of water molecules inside this example voxel here. And it so happens that in the diffusion time that we impart, actually the distance traveled by this water is on the same order of a few microns as the characteristic cell features that we're trying to map here. And so because then the path of these water molecules is influenced by the restrictions and hindrances they experience, the water in some sense probes the microstructure to a length scale that is compatible with cell sizes and this kind of microscopy endeavor. So it does contain information on micron scale features that are way, way below the actual image spatial resolution. And so there are two pillars to the research that we do in my lab. So overall, we want to develop this in vivo non-invasive microstructure quantification, but on the one hand, we focus on these so-called static brain features, where we're really trying to basically map the substrate. So really what the tissue looks like in the living, breathing human. And so here, okay, so here in terms of quantifying tissue microstructure, we've spent quite some time developing models for white matter that are now at the level of really meeting requirements for clinical applications. I will show examples of this today from dementia and schizophrenia studies mainly. And we're also working on developing a gray matter model that is still at the healthy control level, but hopefully soon will be able to be deployed also in human clinical studies. And the other pillar of what we do is to try to exploit dynamic fluctuations of microstructure with brain activity. So in some sense to map the traffic and rather the information that is flowing in the brain as it's undergoing activity, and then how this can be captured by fluctuating microstructure. And so here we're developing diffusion functional MRI as a complement to vascular functional MRI contrasts that are much more widespread, such as BOLD and others that rely on this neurovascular coupling rather than direct neural activity. And so today I will focus most of my talk on this white matter model and applications to dementia and schizophrenia, but I will also spend time to present the gray matter model that is now at the stage of being deployed in the human brain and hopefully soon enough in patients. And then I will also explain a bit about the diffusion functional MRI. But let's get started with the white matter, the first part. So diffusion MRI can be used as a probe of tissue microstructure, but basically what we do, what we record are these measurements with increasing diffusion weighting that we call here as the B value. And so in some sense, it's a very indirect measure because how do we relate then these measurements to the underlying microstructure? So diffusion MRI is very powerful, but it's a very indirect probe. And so then there are different ways to interpret this decaying signal that I've reproduced here. And the first stance that was taken for a while are these so-called signal representations, where really I'm just mathematically describing this decay of the signal with increasing B value without really thinking about what is the underlying tissue that is generating this decay. And the most common signal representation that you're all, I'm sure, extremely familiar with is diffusion tensor, where we stop at this first order approximation here. So when the log of the signal can still roughly be approximated by a linear function. So this is the first term here. And the slope will give you a measure of the diffusion coefficient in this direction. And so if you sample this over enough directions, you can reconstruct the tensor. And then we can reconstruct these scalar maps that everyone, I think, has used at some point. So mean axial radial diffusivity or fractional anisotropy. The representation beyond that can be referred to as kurtosis. And here we're already going beyond this Gaussian approximation regime to also try to capture this curvature of the signal that now makes it distinct from the way the signal would behave in a free water. So if you take a glass of water, you measure the decay, it will follow this straight line. It's always Gaussian and it's always fully captured by this first term here. If you go to biological tissue, this starts to deviate. You have this curvature. And so using kurtosis, we are actually able to capture how non-Gaussian the tissue is and how much it deviates from this linear behavior. And if we sample enough directions, again, we can also reconstruct a full kurtosis tensor, and we can get these scalar maps of mean axial and radial kurtosis, which are then complementary to what the diffusion tensor provides, and can also be used in a clinical setting quite easily. And so DTR or DKI is extremely valuable. So here, of course, it's very sensitive to pathological changes. And DKI has been shown in the literature to really complement very nicely diffusion tensor measurement in a broad range of diseases. So this is not even a comprehensive list. There is now a lot of literature using kurtosis. But the limitation with these signal representations that just mathematically describe the signal decay is that they lack specificity. And so, for example, in a given disease, if you measure an increase in radial diffusivity, what is this due to? What is the underlying biological phenomenon behind it? Is it due to demyelination? So now, for example, the extra-axonal space here would become less tortuous, and this would increase the radial diffusivity. Is it axonal loss? Like I have this patch here of missing axons, but then everything else is fine. Is it some sort of edema? So all of a sudden, my voxel, I have this pocket of free water that increases the overall radial diffusivity at the voxel level. It's basically impossible to go back to this biological mechanism that would actually explain this. And so this is why we push further beyond these signal representations to also propose biophysical models for specific tissue types in the hope to gain specificity and then also much better characterize the pathology in vivo. And so because these biophysical models are tissue specific kind of by design, there is a model for white matter And there is a model for cortical gray matter. Of course, there are diffusion models also for other organs, particularly also for tumor cases, but this is kind of beyond the scope of what I'm presenting today, so I'll just focus on brain roles. And so these models, they come with some kind of expectation that they provide specificity. So we have to be very careful with the assumptions we are making and the robustness, etc. So once we propose a model, we really have to go through a set of questions that build our confidence in this model and in the parameters that we're actually trying to extract from them. And the first one is which microstructure features are truly relevant for the tissue at stake. So we always draw this simplified sketch of the tissue, and we want to make sure that we keep the features that are relevant and that we don't omit any that are really very relevant, but also that we don't add any that actually don't bring anything or make no sense. Then can I sensitize the diffusion MRI signal to those features? So it's one thing to include a parameter in your model. It's another to test whether you can actually have sensitivity to that in your diffusion signal given the acquisition setting. So one prominent example for this is axon diameters. People have tried for years to estimate axon diameters from diffusion MRI until we realized that with the clinical typical gradient amplitude strength that we can reach, actually we have no sensitivity to such short distances and to human axon diameters. And this is why the estimates were sometimes tenfold what you would get from histology, just because the measurement was not suited for actually having sensitivity to axon diameters. Then we have to ask ourselves, can I reliably estimate all the model parameters from the data? This is typically more of a numerical problem. So you have an analytical formula that you have to invert and to fit to your data. And basically, how stable is that fit? What is your accuracy and precision on each of the parameters? And finally, one which I think is really extremely important is how do the microstructure features inform about pathological changes, which is really what we were after in the first place. Okay, so let's now get more specific about this white matter model and then its current applications in the clinic. So it's now fairly well established after quite a large number of years of back and forth and making progress on these models that we can reliably model the white matter as two compartments. And so the first compartment is the intraaxonal compartment that we model as a collection of sticks. So we have some diffusivity here along the axons, and we consider that water inside these axons occupies a relative space in the voxel that is given by this fraction F, which then becomes some kind of axon density or axonal water fraction parameter. And these axons also have some orientation dispersion, one with respect to the other, because they are not, of course, perfectly parallel in real life, if anyone has seen a white matter bundle. And then we have the extraaxonal compartment where we model the diffusivity as Gaussian anisotropic, with one diffusivity rather along the axons and the other perpendicular to them. And so in this model, we just have basically, okay. In this model, we just have 5 parameters to estimate, but nonetheless, it was quite challenging to get them to be estimated, all of them jointly and robustly, but we're really at this stage now. And the most important thing is that each of them can be considered as a specific parameter for some pathological process. So the intraaxonal water fraction is a good marker for axonal loss. The orientation dispersion can be seen to change, for example, during early development when you have pruning, so you're losing aberrant connections, etc. Intraaxonal diffusivity can be specifically sensitive to intraaxonal injury. And then within the extraaxonal diffusivities, the perpendicular one is particularly sensitive to myelination or demyelination, when the parallel one is rather a good marker for inflammation, because if you have a proliferation of glial cells in the extraaxonal space, this will also hinder the diffusion and basically cause this parameter to decrease. So we start to be able to tease apart mechanisms that we cannot do with just typical tensor metrics, which is where we hope the added value is basically. And the important thing about this white matter standard model is that it's really clinically feasible. So what you need in order to estimate it is just a 2-shell protocol. So the minimum you can do is roughly, say, B1000 and another one at about B2000 or 2500 with a certain number of directions along each of these. But once you have this two-shell protocol, you can actually do a lot of things with it. So you can, of course, you estimate your usual diffusion tensor, the kurtosis tensor, and you can estimate this white matter model fully with its 5-parameter. And probably this data would also be quite good for some high-quality tractography. So you can really milk it for a certain number of applications. And we provide open source code to estimate this white matter model that you can readily download and just use to fit your data should you acquire something like this. And then if we compare to the so-called single shell protocol, which is extremely widespread clinically, which basically acquires just one B value along a certain number of directions, unfortunately, with such a protocol, really all you can do is just calculate a diffusion tensor. There's not much more you can do beyond that. I mean, you can do some tractography, but you're kind of missing out on all these possibilities. And in terms of scan time, I mean, now with current acceleration techniques, of course, this single shell protocol has very competitive short scan time, but I would argue that also the multi-shell protocol, we've brought it down now to 5 to 7 minutes, which is something that becomes clinically compatible thanks to the, simultaneous multi-slice acceleration or other kind of newer features on clinical scanners, but it's something that can be actually introduced in a clinical protocol. So if you think of any prospective studies, by all means, try to go with a multi-shell protocol, because it's true that retrospectively, when people come to us with single shell data and say, oh, what can we do with this? Unfortunately, we're kind of stuck and there's not much more that we can do retrospectively. So at least prospectively, I think if we're able to get more and more data sets across different sites that have this multi-shell design, it will really make them, will really enhance the analyses and the richness of what we can extract from these data. And so here's a very compelling example of how these model parameters can be very specific in certain cases of pathology. So this is an example from ischemic stroke published by a group at NYU, where basically we see that in the ischemia region that you see here, the model basically associates the changes to a dramatic increase in the axonal water fraction and an absolutely dramatic drop in the intraaxonal diffusivity specifically, and much more so than the other model parameters, which is actually completely consistent with what we know from ischemic stroke, which is that you have this cellular swelling, so this fraction would go up, and then you have axonal beating. So instead of being a straight tube, it starts to be like pearls, basically. And this really, really slows down the intradiffusivity, which is also what the model captures. So this is the kind of refinement in terms of what we can characterize that we are really after. And so over the past few years, we've worked quite a bit on Alzheimer's disease and schizophrenia. So let me start here with dementia. So this is work that was done while I was a postdoc at NYU. We were looking at the white matter integrity in MCI, so mild cognitive impairment subjects, as compared to their amyloid load. So not so much looking at the diagnosis, but really comparing kind of pathology to pathology or brain tissue state with pathology. And so the amyloid was measured using fluor beta peer PET. And we classified subjects into 3 groups. So they had low amyloid, intermediate amyloid, or high amyloid load. And then we saw how this pathological level of amyloid could be explained or correlated with diffusion metrics in the white matter, such as FA, radial diffusivity, radial kurtosis, and this axonal water fraction from the diffusion model. And what we found that was very interesting, so first of all, for people who still need to be convinced, if you look here, so this is a TBSS analysis of these white matter features as how they correlate with amyloid load in the patients. And so you can see that already the regions where we find differences between amyloid levels and this grouping is actually more widespread. We find clusters of differences in the white matter and radial kurtosis that are not found, for example, just looking at radial diffusivity or FA. So this is something you would get from DTI. And then just going beyond and also using the kurtosis, you see that you also find regions that maybe have more subtle changes in terms of white matter microstructure that is captured by kurtosis, but not so much by the diffusion tensor. So this is also the added value of going to this multi-shell design. But then also looking at the parameters, this was very interesting for us. We found that, for example, the interpretation of the model in terms of intraaxonal water fraction, so the intermediate amyloid levels, so something that you think is kind of early pathology, had higher apparent external water fraction than the amyloid negative, which would really be kind of the controls, right? So we have higher water fraction or external water fraction in the intermediate amyloid as compared to amyloid negative, but also higher in these intermediate levels than the fully amyloid positive. And so if we plot these as bars here, which is maybe more meaningful, so averaging here in the fornix, which is a region of interest that is particularly susceptible to Alzheimer's disease, we have this kind of non-monotonic pattern of diffusion metrics with increasing amyloid load, where also the FA, so it seems first higher in this intermediate level and then dropping as the neurodegeneration really kicks in. And in terms of external water fraction, this also appeared the same way. So slightly higher at the beginning and then dropping as neurodegeneration takes over. And so what happens really in this early stage, because it's very counterintuitive, nobody would expect that you would have more axons as you have intermediate amyloid load, of course. And so the interpretations are possible, glial proliferation at an early stage. If you have some inflammatory response, you'd also have an abundance of glial processes, which are also kind of long and thin and can be somehow similar to axons. So they may be assimilated to some kind of intraxonal water when really it's just glial process proliferation that we're seeing here. Or another hypothesis was a release of iron from degenerating oligodendrocytes. And what this would cause is that your extracellular T2 would become actually shorter. And so this would artificially boost the intraxonal water fraction, because basically these fractions, they're also somehow T2 weighted just because of the spin echo design. So it's not clear actually which mechanism here is dominating, but having this kind of non-monotonic pattern, I think, is interesting to interpret in terms of what the pathology is causing at the microscopic level. And so this is more recent work here from Lausanne, working in collaboration with the Geneva Memory Center. So looking again at cognitively impaired subjects, this time either MCI or Alzheimer's disease versus cognitively unimpaired. And these were diffusion data acquired on a 7 Tesla system, where we compared the diffusion metrics in the white matter, this time with tau load that was estimated from tau PET from floor tau superior. And so here, this is kind of a preliminary study. It's not yet published. So the cohort sizes are extremely low. So I really want you to take this with a grain of salt. The reason I'm showing these results is that they still show kind of the expected patterns, which I found very interesting. So what we did is that we looked at these diffusion metrics in different regions of interest of the Johns Hopkins University atlas. And so we looked at how they correlated with tau load in the subject. And so this is overwhelming, but I'll break it down for you. Basically, here you have the DTI scalars, here are the DKI scalars, and here we have the white matter model that we are estimating. And so these are correlations for different JHU ROIs that we've grouped as some kind of larger families, so you can kind of recognize like internal capsule, corona radiata, etc. And so, of course, if we correct for this, for multiple comparisons with this kind of sample size, nothing survives. But what I thought was interesting, if we look at the model metrics, basically, throughout the ROIs, they really keep a consistent sign of association. So, for example, we have a negative association between tau and intraxonal water fraction and intraxonal diffusivity, whereas the extraxonal diffusivities, which are these two rows here, systematically have a positive correlation with tau load. And this also makes complete sense with what you would expect from the pathology if you have intracellular tau cluttering, right? So these NFTs that are largely intracellular, then this would also, of course, it's known to cause fiber loss, but also would make this intracellular space much more difficult to diffuse, which causes this decrease in intraxonal diffusivity here, when your extracellular space actually becomes freer because you're losing fibers and there's some degeneration, and this would cause these extraxonal diffusivities to increase. So also the sign of change of these model parameters with what is known from tau pathology sort of makes sense. So that's why I thought this was also a nice example to showcase. And now moving on to early psychosis and schizophrenia. So here we're working with a retrospective cohort where the numbers are much, much larger. So we really have a lot of individuals. We have controls, early psychosis and chronic schizophrenic, where the controls are age matched with either of these two groups, when of course the chronic schizophrenic are older typically than the early psychosis ones. So these were data acquired on a 3 Tesla MRI system. And we again used ROI-derived diffusion metrics from the JHU atlas, similarly to before, so DTI and DKI scalars, as well as this white matter model. And what we did beyond just looking at white matter changes in psychosis and schizophrenia kind of standalone, is that because this is a very large study that also acquired all sorts of data, we're also able to link this to blood markers of redox regulation or dysregulation in the disease, and most importantly for us, to brain metabolite concentrations that were estimated using magnetic resonance spectroscopy in a voxel of the frontal lobe. But I will show some of that in the coming slides. So first of all, just from the diffusion perspective and group differences in white matter microstructure, this is a similar carpet plot to before. So here we have the DTI metrics, DKI, and the white matter model. And in this part here, we are comparing early psychosis to matched young healthy controls. And the bottom part is the same, but looking at differences between chronic schizophrenia and elderly healthy controls. And so the first thing that we can see just from the tensor metrics is that we have overall increased diffusivities and decreased kurtosis in the disease, whether it's psychosis or schizophrenia, right? It's the same pattern. And this is, of course, what you would expect from any disease that induces some kind of structural loss or degeneration, etc. You would always have increased diffusivity, so easier to diffuse around, and reduced kurtosis, so reduced complexity, or this non-Gaussian part of the signal. And what was striking also is that in this cohort, the differences in the brain were actually more widespread in early psychosis than in chronic schizophrenia. You see, you have more regions of interest here of your atlas that show differences in an early stage than a later stage. But in terms of the effect size and how pronounced these differences are, of course, the effect size was more pronounced in schizophrenia. So it seems at an early stage, there is some kind of general brain change. People always say, yes, you look at a brain of someone with psychosis, it looks very normal, right? It's hard. These changes are very kind of diffuse and subtle, but nonetheless, we capture them. And as we get to a chronic stage, then it tends to focus to some well-known white matter bundles, but then also with a more dramatic effect size. And so what does the white matter model this time tell us beyond what the diffusion tensor and diffusion kurtosis could tell us? Well, what the model tells us is that basically between controls and patients, the parameters that capture most these white matter changes are the extra-axonal diffusivities that are increased. So to some extent, we also have reduced intra-axonal water fraction, but really mainly the changes seem to be extra-axonal, which is very different from what we saw in stroke, for example, as well as in dementia in relation to tau load. And these changes are, again, kind of make sense with what is known of schizophrenia pathology, that you have myelin defects very early on that may change this extra-axonal environment that we capture with these diffusivities. And then if we look at the effect sizes, actually they are quite similar between the standard model of white matter and the DKI and DTI scalars. So the sensitivity in the model is still there. It's just that the specificity of the interpretation is, in my opinion, largely improved. And so recently we tried to also see how these white matter changes can go in pair with the metabolic changes in the frontal lobe using MR spectroscopy. And so this is now a TBSS analysis relating these white matter microstructure metrics to different levels of metabolite concentrations in the frontal lobe. And remarkably, the model parameters that actually correlated with these metabolite concentrations were again quite limited and specific. We find again this intra-axonal water fraction and the extra-axonal parallel diffusivity that also drove largely group differences with healthy controls. And also there was some specificity in terms of which metabolites we found an association with, in the sense that we mainly and almost exclusively found associations with myoinositol levels. when we really tested also for neuronal metabolites. So we tested for NAA, taurine, glutamate, everything. And the main one really that came was myoinositol and to some extent total choline. So there is specificity in the kind of model metrics that capture this association. There is specificity in which metabolite they are associated with. And also regionally, we see that these clusters of altered white matter also tend to be more frontal, which makes sense with the fact that the metabolite concentration was also quantified in the frontal lobe. So there is also a good spatial association. And so from this association of the extracellular diffusivity with the myoinositol, we can make a link between glial density. So myoinositol is a glial metabolite. Maybe I should have started from this. But so there is a link between glial density, which seems to be reduced in early psychosis, and these white matter extracellular diffusivities that then are increased because of reduced glial density and possibly also effect on oligodendrocytes and white matter integrity. Okay, so this kind of wraps up the white matter part and the applications to disease. So the last part will be much faster, so don't worry. I don't know if there are questions at this point. I don't see anyone actually, but I hope you can hear me. And so if not, I will just keep going just to briefly present you our gray matter model that is kind of ready for clinical translation, and the last part on diffusion functional MRI. So if we now try to develop a similar biophysical model for gray matter, we basically have to start from scratch because it's a different tissue, so there are different assumptions going in there. And so if we think what is different, what makes gray matter much different from a diffusion perspective as compared to white matter, the first thing is that maybe there are more cell bodies in the gray matter that we have to account for as a third compartment, so we can model this as spheres. And the other large difference is that there is a lot less myelin in gray matter than white matter, so maybe now we have to kind of account for some exchange of water between compartments during our diffusion time. Maybe we can no longer consider the water is fully compartmentalized, and we have to also model this exchange term. And accounting for both of these things in a unified analytical model is possible, but actually extremely challenging in terms of fitting all of these parameters. I mean, the model really becomes too unstable and the uncertainty just explodes on some of these parameters, so it kind of doesn't make sense to do it, at least with the currently available data. And so we've established that if we look at our diffusion measurements, there is more signature of exchange in this diffusion signal than there is of the existence of the soma compartment. So unfortunately for now, if we have to choose which one to model versus the other, it seems more important to model the exchange than to model the cell bodies. So this is kind of the stance we've taken when we proposed this NEXI model for neurite exchange imaging. So we've reduced it still to a two-compartment model where we have intraneurite water and extracellular water. And now the parameters are basically suited to what gray matter should be. So we have this neurite or cell process density measure that is a good could be a good marker for loss of processes such as neurites, etc. We have the intraneurite diffusivity, which should be specific to injury. Extracellular diffusion, again, could be a good marker for inflammation. And this new one in the family is the exchange time, where we could use this as a proxy for cell membrane permeability, which can be expected to be highly affected by myelination, but possibly also by some membrane dysfunction. If you think maybe the water pumps are affected or ionic pumps are affected, and then this water exchange also changes from a physiological perspective, basically. And the challenge with this model is that although there are only 4 parameters to estimate, the data acquisition requirements are quite a bit more stringent than for what I previously introduced. So before we have this kind of multi-shell design where we can even get away with just with 2 B values, so you have a fixed diffusion time and just increase your gradient strength here in your regular PGSC diffusion sequence to reach these different shells. So this was before, but now for NEXI, we actually also have to sample different diffusion times. And why is that? So if you think that you need to estimate an exchange time, which is really a dynamic process, it kind of comes to reason that you also have to sample at different diffusion times, because otherwise it's like you take one snapshot, one photography, but then you want to estimate the speed from that. I mean, it doesn't work, right? You need it at different times. And so we have shown in simulations that virtually from a single diffusion time data set, you cannot estimate this exchange time at all. So if you need to access it, then you also have to vary this diffusion time here at the scanner, which makes for quite advanced acquisition protocols for prospective studies. But so this gray matter modeling, we started on a preclinical scanner which had outstanding gradient performance, something that you could never reach on a clinical scanner, and then moving from this kind of high-end hardware, which gave us a lot of flexibility in changing the B values and the diffusion times, we went to less advanced. So we've done collaborations using the Connectome 1 scanner with Cardiff, and also the Connectome 2 scanner more recently with MGH, so deploying NextSeq for the human brain, but on high-end Connectome scanners. And we now also deployed this acquisition and estimation on data acquired on a more regular clinical scanner that we have in our own radiology department here, which has more typical gradient specifications. And you can see that every time we kind of have to compromise on the maximum B value that we have to, that we can go to, and also on the minimum diffusion time that we can sample. And from each of these implementations, we also learned something that helped us improve the model estimation to basically cope with lesser quality data. So already from something Connectome 1 and below, we have to account for Raytian noise floor given the lower signal-to-noise ratio. So this is now included in the model. And also on clinical scanners, because your gradient strength is quite limited, you have to go with broader gradient pulses. So now we also account for this long diffusion gradient duration that takes us away from the narrow pulse approximation. But this is also now accounted for and corrected in the model. So we really improved its implementation and estimation to make it suitable for clinical scanners. And so now we are actually able to estimate these cortical microstructure features in the human brain on a clinical system. And you can see these surface maps here that we compare to the typical myelin map from Glasser et al. That kind of shows the expected pattern that the exchange time here is longer in the central sulcus because you also have more myelination, and also here the neurite density would be higher. So there are spatial patterns that we can kind of make sense of. And then there are spatial patterns typically in the temporal lobe that we have a harder time making sense of for now. And so we're kind of exploring the dependencies of these microstructure features on different things like glial cell density or myelination, etc. And so for the next steps of NextSeq, we want to do more with less. So we really need to reduce the scan time. On the Prisma, it was somewhere around, I think, 35 minutes. But I'll show how we can reduce that in just on this slide. And of course, the longer term goal is to develop a model where we can have both exchange and SOMA estimated jointly, but this will take a bit more effort. And the applications, of course, are numerous and kind of immediate. So of course, the focal lesion kind of pathology is very interesting for us. So multiple sclerosis, TBI, epilepsy. But then down the road, also neuropsychiatric diseases, of course, would be interesting because everyone knows there are also substantial gray matter alterations in dementia or in psychiatric diseases. And so our big question right now for the clinical translation is, can we reduce the protocol without compromise on parameter estimation? And so this is early work by one of my PhD students that he'll be presenting at ICMRM. And the manuscript hopefully is also coming soon. But basically, he used machine learning and SHAP values of importance to do some iterative procedure. And at each cycle, try to rule out or eliminate one measurement. So one combination of B value and diffusion time that contributed least to the estimation. And so he reduced the protocol from the initial number of measurements to just half the number of measurements. So on the clinical system, this would now bring it down to something like 15 or 20 minutes, which is already a bit more acceptable. And most importantly, by doing this intelligently rather than blindly, he was actually able to also keep a very good accuracy and precision of the estimation of these parameters from his optimized sub-protocol as compared to the full protocol, which is very important because we don't want to basically decrease the quality. We want to do similar, but with less fewer measurements and less scan time. But I think this is now going really in the right direction. And so in the few minutes I have left, I just want to walk you a bit through some developments we've done on diffusion functional MRI to try to map the traffic rather than just the substrate. And so this starts from the premise that there are microstructure fluctuations with neural activity. I really want to stress that this has been shown with non-MRI techniques, so just with optical phase coherence imaging, etc. People have really shown that there are movements of the cell membrane, whether in the neuron body here or in the extension, as the action potentials are going through. And this has also been shown in the white matter, right? So even the the myelinated segments of the axons undergo this swelling and displacement as the train of action potentials is going through, rather than the unmyelinated part at the node of rhombia. But so these fluctuations do exist and have been shown in a context that has nothing to do with MRI. And so because diffusion MRI is so sensitive to this microstructure, the question was, can we use diffusion MRI as a functional contrast, particularly in the white matter? So the diffusion functional MRI was proposed by Denis Le Bihan initially quite a few years ago. And the idea was to have an alternative to bold, because you would probably have better temporal and spatial specificity to the activation if you're closer to the neuron firing than the hemodynamic response. But we took a bit of a different stance, because I think in gray matter, bold does a great job, actually, if you ask me. So maybe it makes more sense to use an alternative contrast if you're trying to look at activity that actually flows through the white matter. And this activity in the white matter is really actually a blind spot for most current neuroimaging techniques, because with EEG or MEG, you have very limited sensitivity if you go beyond the cortex, basically. And with bold, well, because you're tied to this neurovascular coupling, your sensitivity in white matter is limited by the vasculature there, which is more scarce and also has a very different response function. And so our goal was really to try to develop diffusion functional MRI for mapping activity in the white matter. But of course, the first challenge is to keep bold contrast out of our measurements. So this is really the ongoing critique of diffusion fMRI since it's been introduced, is that really it's just kind of bold T2 or T2 star in disguise. And so what I do want to stress is that these changes in apparent diffusion coefficients that are related to activity, they have been measured and do exist. First of all, in the absence of vasculature. So here, just looking at a mouse optic nerve, this is older work by Spes and colleagues. I mean, they do measure this drop in ADC in the absence of any vasculature. So if you don't have bold contamination, you do see this change in diffusivity that is correlated with the stimulus. Then groups have done very, very high temporal resolution measurements using, for example, line scanning. And if you have very high temporal resolution, you can also show that this drop in apparent diffusion coefficient actually happens earlier than the bold response. So this is a much faster onset than the delayed bold response. So we do also have a difference in temporality there. And what we really tried to do is to show that with very careful sequence design and acquisition design, in a regular human brain context where you would also have concomitant bold, you can still get ADC time courses that would be minimally affected by bold contributions. And so the way we did this is, so for example, we did studies of visual stimulation here in blocks with a flashing checkerboard. This was done at 3 Tesla. And for bold, of course, we would just use regular. So here we use spin echo to match the diffusion design. So we had spin echo bold, so T2 bold basically with volumes every second. So this would be your bold fMRI neurovascular coupling, typical kind of task fMRI experiment. And for the diffusion design, what we do, and the way you can keep the bold contamination out, is that first of all, you want to calculate an ADC time course. That means you don't just want to go with the diffusion weighted signal, which is also T2 weighted. So this brings in a lot of bold contrast. So we calculate an ADC at every time point by acquiring interleaved two different B values from which you can really calculate an ADC at each time point. Then the second important aspect is in the choice of these two B values from which you calculate the ADCs, you want them to be non-zero. So we chose B values always higher than 200 seconds per millimeter square to directly eliminate most of the direct blood contribution. So above this, basically any active perfusion would just be wiped out, and you're really left more with a tissue diffusion rather than perfusion effects. And the third one is that you want to use a design that has cross-term compensated graded waveforms in order to minimize any possible contributions from susceptibility background gradients that change in a similar pattern that what drives the bold contrast. So there are a few things to be done at the acquisition level, but if you kind of take all these precautions, you can get ADC time courses that are quite immune to bold, and then you can see here that you can really nicely measure this drop and back to baseline, which is much more concomitant with the task than the delayed bold response. And so this was done, so we've shown this pattern using the regular linear encoding, but because our goal was really to use this for the white matter, there was another challenge in the white matter, which was the orientation dependence, basically. So if you think of white matter bundles and there you have microstructural fluctuations with activity, you have most sensitivity if you measure this perpendicular to the axon than along the axon. And in diffusion, I mean, if you do linear encoding, you choose one direction arbitrarily. And so you have spatially varying sensitivity in the brain because the white matter fibers relative to that single direction will kind of vary in orientation. So we moved away from this linear encoding to spherical encoding, which is developed by several groups, mainly by the group in Lund with Filip Stepankiewicz. So basically in one shot, you sensitize to diffusion in all directions of space at once. And so doing this, and again with our visual stimulation paradigm of flashing checkerboards in blocks, so we've shown for the first time that this isotropic ADC-fMRI can actually yield very nice activation clusters along the optic radiation. And those are clusters that are actually not matched in bold fMRI. So of course, bold gives the expected activity in the visual cortex and part of the thalamus here, but it's kind of circumventing this white matter area where there's not a lot of vasculature, whereas the ADC-fMRI basically particularly captures this white matter activity. So this was very, very exciting for us. And we also showed that basically this isotropic encoding compared to the linear one gave the dependence that was less pronounced on the fiber angle, and the ADC change was also kind of independent of the orientation, whereas with the linear encoding, you had a higher percentage of ADC change if the fiber happened to be perpendicular to your direction or not. And this was corroborated also by numerical simulations. So this is kind of a nice complete story that we're now trying to also push forward. And so going beyond task fMRI, is there something that ADC-fMRI can bring to resting state analyses? And here we also looked at this using our ADC-fMRI resting state time courses. And our take was again to try to extend this typical functional connectivity matrix that people would derive like as a region-to-region correlation, and where these nodes would actually usually consist of gray matter areas. So we extended to also include this time white matter nodes. So we've extended the matrix to JHU-ROIs. And so what we found here, comparing the bold fMRI to the ADC-fMRI resting state connectivity, you can see for gray matter to gray matter edges that are represented in green, basically you have a similar picture with the two. But then if you look at gray to white matter edges that are blue, you actually have more of those with ADC than you do with bold. And for white matter edges, which are in pink, they're quite scarce in bold, whereas they're also more pronounced with ADC. So you seem to be able to integrate this white matter activity information in your functional connectivity analysis, which is possibly interesting. And so we looked then at graph metrics derived from these matrices. So again, looking at connections that were gray to gray matter. Between bold and ADC-fMRI, we had kind of similar metrics in node strength, clustering. Okay, maybe some difference here in efficiency, not exactly sure what it means. But in terms of white to white matter graph metrics, actually, for the same one, so node strength, clustering, and efficiency, ADC-fMRI derived was systematically higher than BOLD. So what does this mean? I mean, it doesn't mean that it's better. We don't know how to interpret these graph metrics for now. But what we can say at least is that this ADC-fMRI functional connectivity, including white matter nodes, does yield some kind of robust information that is not present in BOLD. And then what we make of it, I think, is really open for future work. So just to close, I hope I've convinced you that microstructure models of diffusion are very powerful, non-invasive probes. For prospective studies, you really need to adapt the acquisition scheme to your model needs. So go more and more towards multi-shell data sets and, if possible, multiple diffusion times if you have that luxury. And so for white matter, there is very rich information in each of the model parameters, and there's really open access code that you can use to estimate this. And for gray matter, it's very important to model the exchange and eventually, hopefully, also the soma. And we also provide code to estimate this model should you have acquired the necessary data to estimate it. And for the functional part, I mean, there is detectable ADC-fMRI contrast that is clearly distinct from BOLD. So the BOLD mitigation is challenging, but it's not impossible. And I really think the potential for this really lies in white matter fMRI rather than gray matter. I just want to advertise this summer we're organizing a Gordon Research Conference on tissue microstructure imaging. It's in Massachusetts around July, and it's the third time we organize this congress, and it's every 2 years. It's going to be outstanding, and it really brings all modalities together. So not just MRI, but also optical imaging, ultrasound, electron microscopy. So all from kind of with the same goal of looking at biological tissue microstructure, and it's a very rich congress if you're interested to attend. And then I would like to thank my group. So this is a very scary team here, and many collaborators throughout different institutions, funding sources. And yeah, thank you again, Stephanie, for the invitation. And I'd love to hear questions. Thanks.
Thank you so much. That was really wonderful. So much in there and so many questions. There was an active discussion in the chat as well between people. So super exciting. Thank you. I also want to invite the people watching on YouTube to write their questions, and then I'll ask Ileana in the meeting here. So I have like a full page of questions for you. So let me get started until the hands are coming up. And I guess let's start with some methods questions and then move it up a little bit. So you mentioned that you acquired some of your data on a 14 Tesla preclinical scanner, and then you moved it to the connector 1 and 2 for the human data. I was wondering, so in the Netherlands at the moment, they're trying / building a 14T human scanner.
Yes.
Which obviously has a lot of Tesla, but not necessarily the gradients that we need for diffusion imaging. So I was just wondering what your take on this would be, and if you see any possibility of using that for your work as well.
Yeah, I mean, it would be very exciting for sure. I think for diffusion, we do care about gradients somehow almost more than about field strength. So for example, a 3T connectome scanner is kind of an amazing tool because of the very high gradient amplitudes. And then the 3 Tesla does have some advantages with respect to, I mean, more limited distortion or susceptibility effect, etc. So I know people have been quite reserved with doing diffusion even at 7 Tesla for the longest time because we have clinical 7 Tesla systems. We have one here in Geneva. And so recently we actually carried out a few diffusion projects at 7 Tesla in Geneva. And I have to say I was quite pleased with the data because you do have twice the SNR as compared to 3T. And then you could sacrifice that for higher spatial resolution. And it so happens that, well, that system in particular has strong gradients. It's a Terra X. So we kind of had the 7T with relatively strong gradients, which was also nice to exploit. And so this 14 Tesla system in the Netherlands, I mean, you would have a lot of SNR, which are really, we are typically hungry for in diffusion. So I think the SNR aspect may be worth exploring. But then if the gradients are really quite limited, I don't know how that would balance the challenge, all the challenges of scanning a human at 14 Tesla. But definitely worth keeping in mind. I think people have been maybe too shy about diffusion at ultra high field in the human, and
Mm.
actually it's not that bad.
No, I agree. We're getting there, I would say, with the higher.
Okay, well that's exciting, yeah.
Still, like, yeah, 14T, as you mentioned, has some of the problems that we can anticipate already. Great. The other question I had, or one of the other questions was that you mentioned axonal diameter in the beginning,
Mhm.
and that we now have a fairly good estimate of how much we overestimated the results that we had. And it made me think if most of the axon diameter work doesn't really hold anymore.
So axon diameter work done on a moderate gradient system, so anything below connectome,
Mm-hmm.
you would actually be more sensitive. So your axon diameter estimate would be driven by the extraaxonal water. So you have time dependence from water diffusing around these axons, and that gives you a characteristic size of your outer axon diameter. So the axon plus its myelin sheath. And this is something that you can get from acquiring diffusion time dependence on moderate gradient systems. So you have to live with that, but definitely anything that would try to get inner axon diameter on low gradient systems is just quite biased. And I mean, yeah, it does challenge a lot of the existing literature, but this has been shown quite a few years ago already. I think maybe a promising tool that we've also started exploring now is using a surface relaxation theory. So basically acquiring diffusion weighted data. So this is work developed by Mohamed Barakovic and Eric Canales from EPFL. And basically you have heavily diffusion weighted data. So you assume that you suppress your extraaxonal water and you're left with intraaxonal only. And there you acquire it at different echo times, and then you fit T2 decay that could be driven by the axon diameter using surface relaxation theories. And this has produced quite compelling maps of axon diameters in the human brain, even with moderate gradient strength. So going forward on a clinical system, maybe this is a better technique to use if this is a parameter of interest to you.
Fantastic, thank you. Hands are up, so I think Ahmed was first.
Right. Thanks for this very engaging talk. My question is, well, I have several, but I'll start with one, which is primarily related to the white matter bold or ADC bold question. Um, what do you think mechanistically, if I dare use the word, what is the driving factor behind the signal you're measuring with ADC bold? I mean, with regular bold, we're thinking it's more related to the change in oxygen concentration and so on. But with ADC bold, why do you think we are sensitive with ADC but not with regular bold in the white matter?
So just to be clear on the terminology, there is no bold in ADC. So it's
Yeah.
functional MRI using
Yes.
ADC time courses, right? But we're trying actually to stay away from any blood oxygenation effects. And so mechanistically, I mean, we don't have a proof of this. What we've done extensively is to show that the contrast is not vascular. So this is not something I presented today, but we did extensive work with a hypercapnia challenge, so breath hold, and then also recording respiration signals using a cannula to get the end-tidal CO2 also during resting state experiments. And we really showed absolutely no correlation between our ADC time courses and the end-tidal CO2, whether in hypercapnia or normal breathing conditions, when you have very strong association of those with bold, as is quite well known. So that there is really no vascular contribution to these fluctuations. And so what remains is hypothetical because we don't have a direct proof of it. So some of it could be these neuronal or axonal microstructure fluctuations, which have been shown to exist with optical techniques. You also have synaptic boutons that change. I mean, if you look at neural activity dynamically, say under two-photon microscopy, you see that actually the tissue is kind of fluctuating and changing constantly. So this effect, I think, exists. I think what the question that remains for us, because it's not vascular, is whether it's mainly neuronal, or it could also, something we cannot exclude is that it's astrocytic, because the astrocytes are also there a lot to maintain homeostasis. And so they have this ability to swell up to get ions and then shrink back again, etc. So maybe it's actually also astrocytic morphological fluctuations that contribute to our contrast rather than neuronal. But for sure, I mean, we've really looked at this with a very critical eye, and there is no vascular contribution at
Yeah.
moderate field. So if you start to go, maybe also to pick up on what Stephanie was saying, but if you start to go to 14 Tesla, there the susceptibility effects that are at the source of the bold contrast, they are so strong, they kind of like seep back into your ADC measurement no matter how hard you try. But at 3T, it's there's really not much vascular contrast.
Yeah, this kind of makes it it makes more sense in this case why you have your less lag with the ADC-fMRI versus the bold fMRI,
Yes, yeah.
and it's more in line. But if it is more cellular, as in neuronal than it is vascular, do you think in this case that your sensitivity in this domain will highly depend on the system you're measuring? So projection axons will show different types of signal strength or change compared to other. So basically, the question is, if it is entirely neuronal or in large part neuronal, would you expect it to vary systematically with axon diameter or with the support system that these axons have, myelination and so on?
Mhm. This is a great question. We don't have an answer for this. This is something that came up already, saying, I don't know, paper reviews, etc. People asking, what is your response function? Because we know the hemodynamic response function already varies quite a lot, actually, between brain regions and particularly between gray and white matter, etc. When people do GLM, they still use a single HRF, right? And so for us, what is the appropriate diffusion response function? It's still to be established. Given our temporal resolution, to be honest, and how fast these mechanisms are with respect to that, for now, sometimes just using a box function was actually very good. And if you look at the average response we get, kind of smack on the paradigm. But whether there are differences between white and gray matter or between different neuronal types, I mean, this we don't have an answer for. And I'm not sure we could get one with MRI. I think the best thing would be to turn to these optical techniques and then use them to derive models basically of how the tissue is being altered in different cell populations or cortical layers, whatever.
Right, right. Yeah.
Yeah.
And I'm asking this also because of the in the images that you showed, it looked like the ADC contrast ADC fMRI contrast was also more concentrated toward the occipital lobe and along the radiations, which
Yes.
is interesting. Like the fact that it didn't pick up on more anterior signals closer to the LGN side, which I would have also expected. So there's some interaction, it seems, between either the packing density of axons near that region versus in the back, or some of that might have affected it. That's why I'm wondering about this as well.
You are absolutely right to have picked up on this. Um, in principle, the information flows the other way, right? It goes from the LGN to the visual cortex. So we should have seen, if anything, the initial parts of the optic radiation. There, I think, is for now an SNR limitation. So we looked at
Yeah.
SNR maps. So you always have higher SNR closer to your coil element, so to the
Nice.
cortex. And
Hmm.
then, unfortunately, very deep in the brain, we're still not quite there yet because the sensitivity of the technique is more limited than bold for sure.
Hmm.
And so we're just hoping with new developments in like sequences or hardware or denoising, we will be able to also extract information from those midbrain areas. But you're absolutely right. I think the clusters would be there. It's just that they're below sensitivity for now. I'm not sure it has to do with the fiber properties. Maybe it does,
Right,
but
right.
there was such a correlation with the like a distance dependent SNR that we kind of attributed it to that. Yeah.
Yeah. No, thank you. Thank you. This is very
Yeah.
promising.
Thanks.
Eduardo has half a question for you, apparently.
Yeah, sorry. Yeah, I was also curious about the origin of this ADC signal. So just a clarification on this, like you mentioned, it's supposed to be a signal that is not coming from vasculature at all.
Mhm.
By these, do you mean it's not coming from a bolt-like signal, or it's also you're also excluding stuff like small displacements due to vasculature and cardiac activity, for example?
That's also a great question. It's true if you think of like a vasodilation, right? This would also push the tissue and introduce probably some diffusion changes close to the blood vessels that could be attributed to something like that. The answer for that is quite similar to what I said previously, that we tried these hypercapnia challenges where you do have vasodilation from accumulation of CO2, and we didn't see a correlation with that at all. So that's why we think even these vascular deformations, maybe they do cause some diffusion changes in the tissue, but it's not the dominant mechanisms of what we see. And I think the other reason this couldn't really be is that nonetheless, vasculature at the voxel level is quite scarce, right? So you have like maybe 5%. So the water really in proximity to these blood vessels, there's not that much compared to the bulk. So I don't think this would really drive a detectable ADC change. Whereas the neurons, they're much more densely present in like relative population fractions, and this can really cause something measurable for us. But you are right, the vasculature fluctuations, of course, would also affect the
Yeah, no, that's
diffusivity.
true. That really makes sense. Thanks for
Yeah.
the clarification. Yeah.
I would like to bring us back to the clinical study in schizophrenia. Just wait for the slide. But basically, you made this point that you see differences between the younger cohort and the chronic schizophrenic cohort in terms of the variability that you can observe. And I was wondering if you could speculate what might be driving the variability that you observe, if there's different mechanisms, different anatomical features. Yeah. Any speculation onto why we see those differences and why there's more variability in the beginning and then less variability but high effect sizes?
Yes, I don't think I have a very compelling answer for this. One aspect could be that when you're looking at early psychosis population, maybe also the diagnosis is still more diverse. I mean, we don't know how many of these people would actually convert to chronic schizophrenia down the road, or maybe they actually have another underlying psychiatric illness that will kind of clear up or turn up more clearly as they grow to adulthood. So I think there is also more heterogeneity in the early psychosis population than there is in the chronic schizophrenic, which are easier to kind of really put in one box. So this would be more on the differential diagnosis part. And another aspect could be that with repeated psychosis episodes, you actually have sometimes some reinforcements of the same circuits, like you kind of fall back into the same loops or mechanisms. And so maybe in an early stage, it's kind of subtle brain alterations that are more widespread, but as you're accumulating episodes and it's like becoming chronic, it's always the same brain circuits that are engaged, and then the differences there become very pronounced and maybe not so much in the rest of the brain, where maybe you have some compensation mechanism. The last part of what I said is extremely speculative, and I do think the variety of or heterogeneity of diagnosis in early psychosis could actually be a more better explaining these findings. Yeah.
No, it's great. So we look a lot at the variability in the brain, right? And we do see similar patterns. So I was just very curious to see what your explanation was as to what drives them. So it's great to see that we're converging on the results and the theory behind it. I think we grilled you for long enough. Thank you so much for answering all our questions. There's a lot of positive comments in the chat for you. People really
Thank you
enjoy it.
so much.
Thank you so much for being here today and introducing this work. And I'm very happy to see that you're taking the results on the road and show them at HBM soon.
Yes, we are. Yeah.
Hopefully see you then next year.
Okay, thank you once again so much for thanks for all the questions. Honestly, they were really on spot, and I really very much enjoyed the discussion. Thanks for the challenging and thought-provoking questions.
Thank you. Good to see you. Have a good day, everyone. Bye.